Yes, I'm the director of the Center for Smell and Taste at the University of Florida. That's in Gainesville, Florida, which probably most of you have no idea where that is, but it's about 2 hours drive north of Orlando. So we're in striking distance of Disney. So I direct the Center for Smell and Taste, which goes across the entire university. It has about 50 different faculty, as well as students, postdoctoral researchers, and other research staff. It has all sorts of research related to smell and taste, whether it's something very specifically targeted towards anosmia or dysgeusia, whether it might be about flavor perception, about making better tasting fruits and vegetables, insect control, or even just the basic neurobiological mechanisms by which we smell and taste. So we really are quite a diverse group. And as part of that as well, we also have the UF Health Smell Disorders Program. So this is the Associated Health Center, and it's a partnership between the Center for Smell and Taste and the Otolaryngology Department, where rhinologists, particularly Dr. Jeb Justice, runs a clinic that is focused on smell disorders. And we see patients regularly every month for that clinic. And that clinic also helps us to initiate clinically focused research on smell disorders. At the moment, obviously, COVID-related smell disorders are getting a lot of attention.
Mm. Good grief. I know we've talked quite a lot of times before, but that's a lot of different strands you've got coming.
Yeah, I lack focus. Yeah. No.
Yeah.
It's a diverse group, which is a lot of the power of the field, is that people come at it from so many different directions that it gives a lot of new perspectives that can be really valuable for moving the science forward.
So tell us about COVID. Have you seen a big focus on COVID? A lot of people coming through from it in the States?
I mean, it's the numbers vary depending on who's actually been doing the measurement, but it seems that about 50% of adults that acquire COVID-19 also have at least a temporary smell loss. Taste losses, specifically taste loss, as you just very nicely described, is also seen sometime. And even when we didn't talk about chemesthesis, the burn of hot chili peppers or the coolness of mint, that is even affected by COVID as well. But as I'm sure you know, there are a number of people who don't have that short-term transient loss, but that loss persists for weeks, months, or at this point even is ongoing for up to a year, and can also be associated with parosmia and phantosmia, although that is less common. So yes, we're seeing people who have prolonged smell loss coming through the clinic directly because of COVID.
Thank you. Yeah, and Sina's just popped in the chat box. 100% anosmia and ageusia from COVID for over 1 year. Cannot distinguish between any taste, bitter, salt, sweet, sour, etc. I mean, it's a really nasty illness with some really big impacts on people, isn't it?
Yeah, absolutely. And you know, when it first emerged, that the first reports were coming that there was some chemosensory loss associated with COVID, a lot of us, as you were describing very nicely, we've trained you well in the science of smell and taste, is that a lot of times people will conflate a loss of smell with a loss of taste because of that flavor component, the part that allows you to distinguish a lemon from a lime. They're both sour, but obviously there's a big difference in flavor. But there really is evidence that for a subset of people, there is true taste loss. And you'll see video, you can go on YouTube and see videos of people, drinking whole, glass of vinegar or chewing on a jalapeno and just nothing. And no response at all. And it's devastating. It's devastating.
Okay, well, it's fantastic you're there to be able to be looking into it. Here in the UK, I every time I've sort of talked to Carl Philpott and see that he's the sort of still fairly fresh-faced, and and here he was coming in as a doctor and managing to open up the first smell centre here in the UK. Was it similar in the US as well, that you're breaking new ground for one of our senses. I mean, it still seems extraordinary between the two.
It is true. I mean, the University of Florida clinic is not the only one in the US, but there are only a handful, really. They're starting to become, I mean, one of the silver linings of COVID is that obviously it's bringing a huge amount of attention to these disorders. And that is increasing the education of otolaryngologists, who are primarily the people who would see in specialty clinics those who have a smell and taste loss. And so there are more who are becoming better versed in the diversity of sensory deficits that can happen with smell or taste, and trying to, unfortunately, as we all know, that the treatments are few and far between, but certainly providing better support and diagnosis has been happening.
Fantastic. Thank you. Well, so the title we've got today is, tell us, Gene Therapies and, sorry, it was a long title, I did forget it.
Yeah, stem cell, yeah. So let me go ahead. I can't share my screen. Dan, could you?
Oh, I disabled it because somebody else was as well. Let me actually make you a co-host, would be easier, and then you can share.
Okay. There we go. Looking good.
So just a reminder, as we go through, any questions, any thoughts you have, do pop them into the chat box. We're going to have plenty of spaces for Q&A throughout our time together today.
Yes, and I've actually worked in a few spots throughout the talk to force myself to pause and give some time for questions rather than holding them off to the end. So we'll have some time to talk. What I thought would be useful to do is give you a little insight into some of the things that the research community is starting to focus on in their effort to try to get to effective therapies. And right now, the only therapies that we really have that are effective are related to conductive smell loss. That is chronic rhinosinusitis with nasal polyps, where surgical treatments or steroids might be useful. Some other sort of surgical approaches that can be that can resolve the issue for a lot of patients. But certainly for post-viral smell loss after head trauma and these associated types of chemosensory disorders, we don't have many real options. A couple of years ago, the major research centers in the United States, and I'm going to skip over these first two slides, which we already talked about, our smell disorders program in the center. The major research centers in the United States got together at the behest of the National Institutes of Health, which is our major medical funding agency for the federal government, and came together to explore approaches that might be most promising for developing therapies. And so I'll talk briefly about that meeting, but also the white paper that came out of it that is now published and freely available. I believe Fifth Sense has it up on the website, but if not, we can make sure that that's available to everybody. And then I'm going to give you some snapshots of some ongoing research from a few laboratories, some at Florida, but some at other institutions, that are exploring sort of cutting edge therapeutic approaches using gene therapy, stem cell therapy, and or basically the equivalent of cochlear implants to try to restore olfactory function. So the question of how do we get to treatments has been a multifaceted problem for our field for a long time. Part of that is due to just the few people doing research that is directly related to that clinical translational research that can lead to that sort of development. It's also been a lack of understanding by the broader community, including by government funding agencies that supply the money that is needed for biomedical research, to really prioritize smell and taste disorders for funding and for support. Frankly, because the view was it's not as important as a lot of other health issues. Something I think this audience knows is certainly not the case. Well, as part of this, in consultation with a group in the National Institutes of Health, and this is called the National Institutes of Health. It's multiple institutes that focus on different aspects of health. So we have a National Eye Institute, we have a National Cancer Institute, National Institute on Drug Abuse. In this case, it's the National Institute on Deafness and Other Communication Disorders. And this is the institute that oversees research in hearing, balance, speech, language, smell, and taste. And so this is our home. And they came to us with the idea, collaboration with the Monell Center in Philadelphia, the Rocky Mountain Taste and Smell Center in Denver, and the Center for Smell and Taste at the University of Florida, about bringing together a group of scientists within the field who were trying to develop these cutting edge types of approaches, as well as scientists from other fields, other sensory systems that might be able to give us some insight. So this, we never came up with a better name, but Identifying Treatments for Taste and Smell Disorders was the name of the meeting, the ITTSD. And this meeting sorry?
It does the job. Can I just sure. check in with you quickly, because we've got a couple of so we've got quite a few people here who've got congenital anosmia, so that's just coming up. So somebody's just said you're using the word restoring. So, but this could also be for people with congenital as well who've never experienced this sense of smell.
Absolutely. That's sloppy jargon on my part. And actually, the gene therapy we're going to be talking about is specifically in the context of congenital anosmia.
Fantastic.
So there'll be, and that's probably where gene therapy is going to have its earliest promise, while stem cell therapy may be the better route initially for acquired anosmia and other disorders.
Fantastic. And so that was my next question was, and Charlie, I think that answers your question, and Stephen will get to that piece. Are you going to cover off what, for those who have acquired smell loss, as to how they acquired it? Does it make a difference for what you're talking about? And is that something you're going to cover?
Certainly, it's something we can certainly talk about, and it probably will make a difference, because exactly what part of the system is damaged is going to influence how you're going to target the system to try to get it functional again. So we can certainly talk about that as we go through.
Fantastic.
This meeting, the ITTSD meeting, when we set it up, it was a 2-day conference. The second day was primarily clinicians speaking, as well as we had patients that came from that largely that geographic area, that some had some presentations, but also sort of participating in discussion. And I'm not going to talk about that part. I'm going to talk about what happened on the first day, which was really targeted to stem cell and gene therapies. And what you can see by this list, and I'm just going to highlight a couple people. So, for example, Linda Barlow in the stem cell therapy, and Peiwa Zhang there as well, are two researchers have long been interested in stem cells within taste buds, and how understanding how these stem cells give rise to mature taste sensitive cells in the taste bud would be a way to restore taste function when things are damaged. And the damage could be, Linda Barlow is very interested in damage that accompanies cancer therapies, chemotherapy, radiation therapy that can disrupt. And for example, Peiwa Zhang has been doing a lot in trying to basically build taste buds in a dish, so they can really study how these cells give rise to other cells and be able to turn them on and turn them off. But I will talk about, you see Bradley Goldstein at Olfactory Stem Cells in the clinic. I'm going to talk specifically about some of his work, where he's been able to do in preclinical studies actually take stem cells, put them into an animal that has been rendered hyposmic, and restore some of that function. In the gene therapy realm, I particularly want to, before I talk about Jeff Martin's work, which is some of the gene therapy work I'll discuss in the context of congenital anosmia, is mention two researchers, Shannon Boy and Jeff Holt. These individuals come from different sensory fields. Jeff Holt is very interested in function and dysfunction with hearing. And Shannon Boy, in the same case with retinal disease. In both cases, that they have been leaders in developing gene-based therapies for restoring function. And this has actually been done now. It's been done in a couple of different cases in humans with retinal disease, where actually application of viral-based gene therapies can rescue a dysfunctional gene, create a functional gene that can then do the job it's supposed to do in the tissue and allow that tissue to restore relatively normal function, basically recover sight. So these are the types of studies in other sensory systems that are inspiring those in the smell and taste world to try to follow the same paths. All of the researchers that were involved in this group got together and put together a paper identifying treatments for taste and smell disorders, gaps and opportunities. This was published finally last year in the journal Chemical Senses. Disclosure, I'm the editor-in-chief of that journal. This is freely available and will, if it isn't already, will be posted on the Fifth Sense website. But there's also the URL for it there at the bottom of the slide. This is discussed not only the science that was presented at this particular meeting, but also put forth some priority areas that we thought needed to be emphasized going forward. And these were both research priorities and community priorities. And one thing to remember, that since this was a US-based effort, some of these might be more relevant in the US than they are here. For example, number 6, encourage the formation of patient advocacy groups, something like Fifth Sense, that has really been progressing well in the UK over the last many years. The ones in the US there are a couple that are only in their very nascent forms. This is important not only to lend support to individuals, that peer support, and provide education and information, but it's also critical if one is to lobby funding agencies, lobby government agencies to put more resources towards this problem. And I can talk till I'm blue in the face, but to a congressman or whatnot about how this is important, it's not going to be nearly as meaningful as a group of people who are actually directly affected by these disorders. So that's something we certainly emphasize. But what I'm going to focus on today are really what came in points 1, 2, and 7, which all surround trying to get better and more research and researchers in the areas of stem cell chemosensory biology. Now, this is all very relevant for both smell and taste. Oh, sorry?
Oh, no.
So this is all very relevant for both smell and taste, but quite frankly, the research has progressed much further in smell. And because of the time we have, I'm not going to get into any of the taste research, but I just did want to emphasize here that indeed taste buds have stem cells. These are tissues that are constantly regenerating throughout your life. And you can imagine why that is because they are constantly being exposed to toxins and viruses and heat and other things that could damage them as you're eating or you're inhaling. And so these cells, these long spindle-shaped cells that are in this garlic-shaped cluster at the top, the green, the purple, and the brownish ones, these are the cells that are responsible for sensing different tastes, sugars and acids and salts and such, and then communicating via the nervous system that taste information back to the brain. But those light purple cells and those blue cells at the very bottom, the ones the arrows are coming out of, these are what we call stem cells and progenitor cells. These are ones that have the possibility of differentiating, of changing their identity into these sensory cells. And so there's this constantly churning population of stem cells. And if one could imagine that you lose those mature sensory cells at the top, and you can no longer sense taste, but you still have the stem cells that you can somehow target to turn them on again to make those new adult cells. That would be very powerful. Or if you're lacking in the stem cells, but you could grow them in a dish and then transfer them in to the taste bud area and to bring about this birth of new sensory cells. That would also be quite a powerful structure. So those are the types of things that people are thinking about, but partly because taste buds are so hard to get to, they're embedded in your tongue and your soft palate and such, that hasn't really gotten as far as we have with the sense of smell. And so at the very beginning, you saw how the olfactory apparatus, the cells that are responsible for detecting odors, are up there in the roof of the nasal cavity. And this is just a higher magnification cartoon of that area. So if we see at the bottom of the slide, you see in the sort of the yellow, the blue, the reddish cells, these are what we call olfactory sensory neurons. At their tips, where they have the cilia that projects into the mucus that overlies this tissue, or underlies it, I guess, since we're upside down. That's where all the molecules are that respond to odors, that can detect those odors, and that can initiate an electrochemical signal that is sent back up in the picture through the cranium. So the cribriform plate is the part of the skull there, and into the brain, and specifically goes into this area called the olfactory bulb. That's the first part of the brain where odor information is being processed. And then it makes connections with other nerve cells that take you back to other parts of the brain. There are a couple other cell populations I want to emphasize here. These light purples, rectangular cells that you see throughout, these are known as sustentacular cells, or supporting cells. These supporting cells provide the proper environment for the tissue to function. And one of the best illustrations of how important they are is that we actually think that those are the cells that are targeted by the SARS-CoV-2 virus. The virus causing COVID and that disrupting olfactory function, we think that the virus is actually hitting the supporting cells, either killing them or so disrupting their function that they damage the ability of the sensory neurons to respond to odors. So the sensory cells themselves don't seem to be sensitive to the virus, aren't being infected by the virus, but their environment's being disrupted because these supporting cells are being damaged. Then the last group of cells, and another one we'll focus on, the basal cells, so those little squares you see there. The basal cells are, these are cells that contain that stem cell population. By stem cell, what I mean is it's a cell doesn't have really it doesn't look like a neuron or supporting cell or any blood cell or anything else, but it has the capacity to change into any of those. So once it divides and turns on certain genes, it can become a supporting cell, or it can become a sensory neuron. And there are a variety of different versions of these stem cells and progenitor cells within the basal cell population, but these are going to be the ones that are targeted in stem cell therapies. So I guess actually before I go on, Tom, are there any questions you think would be good to address right at the moment?
There are a few coming through. We had one from, Avian has asked, very hard question that appreciates to answer. Is there an estimation when we're looking at this being turned into treatment?
That is a hard question, and I will get back to that, but I think it'll be more informative after I show you where the research is. So fantastic. If I forget, Tom, please remind me to get back to that, but that is an important question.
Deborah's asking, any stats for how many COVID patients fully recover based on the research you've done at this level?
It's not our research specifically, but just from research in the field, what we see is that, the numbers vary depending on how it's being assessed, but it's looking like at about 2 months out from the initial loss or from the initial COVID infection, 80 to 90% of individuals will have had substantial recovery, if not complete recovery. It's seeming to be somewhere in the 5 to 10% range has very prolonged loss. And fortunately, since we're still so new in this disease, it's only been around for less than a year and a half, we don't know how many of those we could really categorize as permanent versus prolonged, because certainly if we extrapolate from what we see with other post-viral anosmia and hyposmia, we do see people that recover significant amount of function even a year or 2 years or more out. So it's hard to say that anybody has permanent loss at this point, but there certainly seems to be about 5, maybe 10% of people have prolonged loss. And that's similar to what we would see with other post-viral anosmia and hyposmia.
And coming back to the question I asked earlier then, so is this stem cell research going to be applicable to post-COVID and post-viral if it involves damage?
Yeah, so that would be the hope. Until enough, unfortunately, post-mortem individuals are examined to actually understand exactly what damage was done. We're strapping a lot of this is we're inferring from different experiments and different understanding how the virus works. But until we really see that, are stem cells still there in people who have had prolonged loss? If they are, then that suggests that perhaps you can stimulate them in some way to replenish the epithelium. If they aren't there, if they're also ruled out or knocked out by this disease, then there might be need to be engrafts of stem cells from other individuals or from cell lines that could potentially do that. And I'll show you some research that sort of implicates that as a possibility.
Thank you. So a few more specific ones. Are these true stem cells, or are they only able to come olfactory cells? Question there from Charlie.
Stem cell is a term that is contextual. So it is not, they are multipotent stem cells in that they can give rise to multiple types of cells, but they are not pluripotent stem cells in that they cannot give rise, at least if you just take them out without treating them in some way, they can't give rise to every type of cell in the body.
Gotcha.
Although there are ways that people can treat the cells with certain molecular factors in a dish that can sort of reset them, but then they'd be there. So these are ones that are able to go in multiple directions within the olfactory system. And as they go through that path, they become more and more restricted on the options that they have.
Got you. I think I'm saying I got you very confidently, but it's very comfort. I love the zooming in, the level of detail of each one. A question from Keith saying, which cells excrete mucus and what's the significance of mucus?
So the mucus secreting cells are actually not shown in this cartoon. They're glands within the olfactory epithelium called Bowman's glands. That's BOWMAN''S. And these glands secrete the mucus, and the mucus is probably having a variety of purposes for the tissue. It's protective. It allows these very fine olfactory cilia to have a somewhat protected environment to sit. It also means that odors that are going to approach the cells have to sort of dissolve into that mucus and to become accessible. And there can be other proteins that we call odorant binding proteins that may contribute to that process, or shepherding the odors in and out, because you also want odors to go away. You don't want to have them sitting there for your entire life.
Of course, yeah.
Yeah, and that mucus has all sorts of different ions, minerals that are other factors that are there that contribute to the ability of the tissue to function.
Fantastic, thank you. And last question for now, Mike, is any of the stem cell and gene therapy research happening in the UK and Europe?
Not that I know of.
That's the second part of his question. If not, why not?
I think it's a combination of funding, of people that are interested in these particular systems. Mm-hmm. The smell and taste research community is much smaller than the community that studies vision or hearing, for example. So just the numbers are, in the UK, there are not many smell and taste researchers that are working at, certainly not ones working at these sort of basic mechanisms one needs to do to focus on in order to develop these kinds of therapies. There are certainly a number of researchers. Carl Philpott, you mentioned one, who are very much on the clinical side or on the sensory side.
Yeah, thank you. Mm-hmm. Great.
Okay.
Nice.
So as we move on, I want you to focus, we'll talk about predominantly these two different groups of cells, the olfactory sensory neurons, those long nerve cells that carry odor information from the nose back to the brain, and then what are listed here is basal cells, these stem cells and progenitor cells that are important. Now, our first view, I'm actually going to take this cartoon, it's going to be flipped 9 degrees. So you're going to be looking down on the surface, on the top surface of the supporting cells, and you're going to have one olfactory sensory neuron. Go forward. Come on. There we go. On the left there. And so that's what it looks like if you were to look at the surface of the olfactory epithelium. So all the short little protrusions, the fuzzy bits, those are what are called microvilli. Those are the tips of the supporting cells, the sustentacular cells. And right there in the center is a single olfactory sensory neuron. They're usually a little more denser than this, but this was one selected because it's just a solitary one sitting there. It has an end in that knob, and then it has a bunch of these long cilia. And those are sitting out there. They don't move. They just sit and float in the mucus. And they are where odor detection first happens. They have receptors and other specialized proteins that allow these cells to recognize odors. Now, on the right, I'm showing a colleague, Jeff Martins, there in the white shirt, and some members of his lab that have been interested in quite a long time in those olfactory cilia. And one reason they are interested in that structure is because it is so absolutely essential for olfactory cell function. So they've been studying these in a context of a group of diseases known as Ciliopathies. These are largely genetic diseases that have, or genetic syndromes that have individual or multiple genes mutated that result in the malformation or the complete lack of cilia. And those cilia, there are other types of cilia that are affected in the kidney and in the brain and in other tissues, but it's particularly in the nose where their work is function. So Bardet-Biedl syndrome is one of these. Joubert syndrome is another one, but these are common. So these are not particularly common genetic disorders, but these individuals do have anosmia. They do lack the ability to smell. So the Martins lab has created mouse models of these human genetic diseases and focused on the sense of smell and trying to rescue it. So here is a cartoon of how one of the papers from them. So they're, excuse me, on, ignore the top and the middle, just look at the bottom two right panels, the middle and the right one. So that is a micrograph of a single olfactory sensory neuron there in the center with really extensive cilia. And you can see how when it's kept intact like that, there are just tons of them, they're really long. And then there are a couple of other ones there to the side with the arrow where the cilia seem to be missing or really truncated, they're really short. In a particular disease state, and so like is seen on the left-hand picture, you may have a lack of these cilia, and the goal here is to try to rescue that, to restore the cilia and then restore the function by putting the proper gene back in, the functional gene back in. And this is done in a very simplistic way, is that they take a pipette filled with a virus that carries the proper gene and spritz it into the nose. And this is done in mice, so you can make these genetic manipulations. And so you just spritz this virus in the nose. And just like I'm sure a lot of you have heard that many of the COVID vaccines that are out right now use a innocuous virus to transmit the material you need for that vaccine into the body. In this case, that's what the virus is doing as well. It's basically just a shuttle to take that repaired gene and to inject it into olfactory sensory neurons and try to rescue them. So what happens if we actually look at how this affects the sense of smell in these animals? And we're going to look in three different ways. We're going to look at physiological measurements. So some of you may have had an EKG or an EEG, electroencephalogram, an electrocardiogram, at some point to measure the electrical activity of your brain in an EEG or your heart in an EKG. This is an EOG, an electro-olfactogram, and is measuring the electrical activity that is happening at with those olfactory sensory neurons. And we can tell, if you look there in panel E, is the best way to look at it, that normally if you give the animal, you spritz on different increasing concentrations of odors, you get a bigger and bigger signal. The dip gets bigger before it recovers, showing that's a normal odor response in these animals, and these are normal animals. However, in that second group, the IFT88 OSN-KO, which means knockout, that one particular gene called IFT88 has been deleted in this animal. So it doesn't have cilia and it doesn't have that function. Those animals basically don't respond to odors. There's no electrical response. However, if you put the functional version of IFT88 back into the nose using this viral gene therapy approach, you can see that there's a rescue and the response returns to nearly normal. There on the right side, there on the right side, that's just showing the group data, showing if you did it with a bunch of different animals. The black is the normal animal, the gray is the animal that has the disrupted olfactory sensory neurons, and the blue are the ones where the genetic rescue has occurred. And you see the responses to all these different types of odors and odors of different concentrations have been returned. So this animal, from at least a physiological measure, is getting normal sense of smell. How about if we look at the way it connects to the brain? Because just because you can detect something in the nose, if that information can't get back to the brain, it doesn't matter. So this is a special way of tracing how those sensory neurons go from the nose to the brain. What you're seeing there is the olfactory bulb, and you see these long trails of blue little lines. Those are actually individual nerve cell axons, the processes they send from the nose into the brain. And they come together at that arrowhead in a single point. And that's the way it normally works. And these are all olfactory sensory neurons that are expressing the same olfactory receptor, so they respond to the same odor. On the left, you see that they're all coalescing together into that one spot. In the middle, however, they're having a harder time finding the same spot, and they're going to multiple ones. So you see the two arrowheads and the higher mag version there in the inset. But then with the rescue, once again, they're going back to all together to the same place. So this is important. So you're able to, the cells are now able to respond to odors like they weren't before, and they're able to make connections back to the brain like they weren't before. But the real kicker is, are those connections meaningful to the animal? Is the animal now perceiving odors in a meaningful way? And so the last way to test that is through behavior. In this case, I just want you to look at these four sort of X-like structures. What this represents is a chamber where a mouse is put, and there are four different corners, and each corner has a vent, one of which will give an odor, the other three will give just a stream of air. And so in this case, it's the red quadrant that gives the odor. And in the center, there is a vacuum that pulls the odor in. So it's coming from the four corners, and then it's getting pulled into the center. And so there's a gradient in each direction. And the mouse, likes to explore new odors. So if he hasn't seen them before, you put him in the center, and he's going to wander all around and check in all the corners. But as you can see in that left-hand panel for the animal is called the WT, or wild type, the normal animal, they go predominantly to the corner with the odor in it. They spend much more time there. In the animal where the gene's been disrupted, where there's no longer a functional olfactory system, they spend pretty much equal time in all corners. And then in the rescue, they once again are able to find the odor. And that information is all put together in the right-hand panel, where you see that the response is almost back to normal. So this therapy, this gene replacement therapy for a single gene congenital anosmia, has been effective at the level of the responses in the nose, at the level of restoring connections to the brain, and of making these connections meaningful to the animal such that they can make the right decision about whether to investigate an odor or not. So this is a good place to stop for some questions.
Yeah, so in the context of this particular one, sorry, I can't remember, is it IFT or LFT?
It's IFT.
IFT, yeah. So does that correspond to a disorder in humans as well, or is that a mice only one?
So this would is one of the Bardet beetle syndrome genes. Got it. So are related to that. So it is, the gene is called interflagellar transport 88. I think it's 88 kilodaltons in size. Yeah. Mouse people don't name things fun like some others do. People who work on fruit flies they have some very creative names. So this is a gene that is found in some forms of human disease, but it's not this particular gene that's the important thing. The Martin's lab has now done this rescue with several different genes that disrupt the olfactory cilia and been able to rescue them similarly. So the key thing is, so that shows that it's not just this one, only people that have this one mutation would be potentially treatable, is that if this is able to move towards a clinically useful therapy, then you can imagine variations of ones that address all sorts of different types of genes. And they wouldn't only have to be ciliopathies. There's evidence for people who have mutations in different genes that are involved in coupling the detection of odors to that electrical signal that the nerve cell has to send. And so they can't, their cell breaks down in that connection. There are other ones that have been identified that are those simple gene mutations that would be very ripe for gene therapy. And then the other place that gene therapy could be useful is with those stem cells. If you have a population of stem cells that seems to just be dormant, is not dividing to make and replenish the epithelium, providing via gene therapy basically an on switch could help to stimulate that progression and help to recover a depleted tissue.
Got it. So it's at its most basic, it's a proof of concept that you can introduce a gene therapy in this method with this method using a
Right,
virus.
right.
Yeah.
And so now, so back to the question that came a little earlier about timing. What needs to happen to get this into human clinical trials?
Yeah.
So there are a few key issues. Obviously, one needs to address safety. And so there are different ways one goes through that. One needs to decide the exact best virus to carry these genes to the nose that will not only be safe, but also effective and target the right cells. Because for this particular thing, if you target the supporting cells, it won't matter because that's not where the gene's needed. It's in the sensory neurons. Similarly, if you had something wrong with stem cells, you need to target it specifically. So finding the exact right version of the virus, these are called serotypes, is important. And then one needs to work on things like dosage and frequency. The olfactory epithelium turns over every 30, 60 days. You're constantly making new olfactory sensory neurons in a normal nose. So do you need to do this once a month, once every 6 months, daily? Those types of things all will need to be investigated prior, and largely done in animal models, prior to doing those initial safety studies and efficacy studies in humans. You're muted.
So a couple of quick questions from Charlie, our resident geneticist, who's asking, by the sounds of it, you start with the genetic sequencing and then apply the right gene therapy for a patient. And she also asks, do you think we could get to the point we could diagnose congenital anosmia with a genome sequencing rather than scans?
Yes, to both. I mean, genome sequencing, especially what we call exome sequencing, which is looking at the parts of the genome that actually encode the proteins that you're interested in making, is becoming much more affordable. And it's not going to be long before that's going to be a regular part of probably most people's regular health care of understanding a genetic risk and genetic, what they call personalized medicine of, certain medications, especially we see this a lot of cancer therapy, where one medication might be good if your tumor has a particular genetic variant, but it might be completely useless if it has another. These types of things are going to be necessary. At this point, we still don't have a good sense of how common what we call non-syndromic congenital, non-syndromic gene mutation dependent congenital anosmia is. Because certainly congenital anosmia can result from not just genetic differences, but could result from injuries during birth. I mean, it used to be when their forceps were used more often, that was probably a cause of physical disruption of the olfactory nerve when the skull was reshaped. Yeah, please.
No, I was also going to say that I knowing and being friends with Duncan and knowing how head trauma can cause anosmia, I just, my heart was constantly stopping when my kids were toddlers. Boof. Yeah. How did they get through that with a?
And there certainly are probably kids that cannot, that by the time they are able to really talk about that and really realize something about the sense of smell, they can't remember that they ever could when they were 1 year old or something like that. So in this particular case, we're talking about people who have specific gene mutations, and if you could identify those, then you could tailor the therapy to that. Absolutely. But using gene therapy to get at the stem cells may be another way that is more broadly useful for a variety of different conditions, no matter how it was, what the initial cause was.
Great. Thank you. Just as a sort of a general thought, because some sort of bit of chat, in terms of sort of prioritization of this research. What's your, and I know as a community we've read, like, we know this is really important. In terms of the wider field of how this gets priorities inside medical research, what's your own sort of personal view? How do you put that across when you're talking to other researchers?
What I tend to do, and I actually did this fairly recently in an op-ed in USA Today, which for those of you who travel to the United States may recognize is the paper that gets shoved under your door at hotels. But it's sort of a national paper. But making the case in two ways. One is this is not a rare set of problems. Mm. Smell and taste disorders are incredibly common. And one comparison I made, that if we look at people 40 and over, and we compare the prevalence of clinically meaningful, so hyposmia, anosmia, that is something's not treatable, so ruling out, say, nasal polyps, blocking smell, and compare that to non-treatable or non-correctable visual deficits, it's about fourfold more common as smell disorders are. So that alone sort of, I think, gives people pause in realizing that this is a very common thing. And as I think all of you know even better than I, is that, you know, no one's going to know you can't smell or that you have parosmia or something else unless you tell them. You don't have a service dog that might, or a cochlear implant, or use sign language or something else that may signal to the world that you are sensing it differently. So it's in that way, it's sort of silent to the broader population. So just educating people about how common it is important. The other is emphasizing that it's not just an inconvenience, that that these disorders have real health consequences, not only for safety and diet, nutrition, things like that, but also for mental health and quality of life and interacting with others in your environment. That these are not inconsequential things. And if the cause was something other than a loss of smell, it'd be taken a lot more seriously generally, I think, by the health professions. So I think educating people about the real impact it has on people and the prevalence are two critical messages to send.
Yeah. Fantastic, thank you. And of course, we talk a lot about prioritization of funding, but of course, there are hundreds of thousands of areas of medical research happening concurrently around the world, with money flows coming in and out of them accordingly. So, great. Oh, sorry.
Oh no, I was going to say, I don't think I don't think it's probably serves us, any of us, well to say, well, this is more important than disease X or less important. I mean, and the truth of the matter is the way science works, a lot of times, I mean, just as we talked, the whole basis of that meeting I started talking about at the beginning, we're taking the lead a lot from what's happening in vision science and hearing science, techniques that they're developing there that we're able to translate over here, and it's going to go the other way as well. So so that cross-pollination is important.
Fascinating. Just noticing time, Steve, and I don't know whereabouts you are in your
I can go. Yeah,
presentation.
I know we've had it. We can speed it up, and I'll make it very quick to wrap this out. I just want to highlight two other people. These are neither of which are at the University of Florida. Brad Goldstein at Duke University, who's focused on stem cells, and I'm just going to quickly go through just to remind you the stem cells, these basal cells that can give rise to a variety of different types. What he has found, and this is again in mice, that he's been able to create a strategy to isolate stem cells from a mouse. And I won't go through all this, so just trust me on that. But those stem cells, he can then just put them back into a mouse using the same type of spritz it up the nose. In this case, he calls it droplet engraftment, where the mouse sits there and sniffs it, much like, you know, your pet dog likes to go sniff the urine of his friend down the street. The cells are taken up into the nose and they become incorporated into the olfactory epithelium. And if you label these cells with a a fluorescent protein, so they all, in this case, glow green, you can track them and know which ones came originally from the mouse and which ones came from the graft. And this just shows they've been able to make a hyposmic mouse by, again, getting rid of the cilia. But this is the key one to focus on here, is now taking these mice that are hyposmic. They have some functional sensory neurons, but others that have been made non-functional by this genetic manipulation. And then you graft in stem cells from a second mouse, and these stem cells are green. And what you can see, if you look in panel B there, you can see that they've not only started to get into the epithelium, but they're starting to distribute themselves, and they're becoming sensory neurons. And then on the right, I showed you these electroencephalograms, the EOGs, these electrophysiological measures of olfactory function in the nose. The control animal, that's the one that started. The IH animal is the one that was made hypoxic, so the black on the right-hand side of panel G. And then the red in the middle, you see a partial recovery of that EOG response. And then they did an odor avoidance task, so they used an odor the mouse typically would want to go away from, rather than one that would be attractive and want to go to, like I showed you for the gene therapy. You see, again, the control mice avoid it. The hyposmic mice don't seem to really care one way or the other. And then the ones with the stem cell graft start avoiding it again, indicating that they have reacquired the ability to smell. And the last slide is just showing that these grafts do look like they contain neuronal genes. But let me just, before we stop for questions, just 2 more slides and give you, no promise, stem cells and gene therapy, but there's one other type of approach that is in its early stages, but has also been inspired by what's happening in other sensory systems, and that is using some sort of electrical stimulation to recapitulate the input in some way. So we know this works really well with cochlear implants, and a cochlear implant is basically an external sensor that senses the vibrations in the air, what sound is, and then conducts that across the skin and then via a wire into the cochlea, the part of the ear that is responsible for detecting those sounds. And so this electrode that's going into the cochlea carries that sound information that it's acquiring from its sensors and gives it to the nerves that then carry sound information back to the brain. And it has now gotten to the point with a number of different electrodes that are inserted this way, that the quality of speech recognition, for example, is really, really good for these individuals. And it can be really a really powerful rescuing treatment for those who are deaf and want to go down this road. Could we do something similar for smell? Now, of course, we're not looking at something like the head of a drum that is just getting sound waves and bounces, pressure. We have to have some sort of sensor that could recognize thousands of different chemicals in our environment and track them separately in a way that makes sense. And that's where a lot of the basic science is still really important. But in very initial studies in rats and in humans, there is some support that this kind of approach could work. So on the left, from Rich Costanzo's lab at Virginia Commonwealth University, they took an electrode, a 7-channel electrode in this case, and put it associated with the olfactory bulb of the rat. So this is the brain structure that receives information from the nose. And then what that square on the top is the sensor array, and they're measuring from that one red spot. And what they see is, in response to electrical stimulation at the bottom of the bulb, and along the nerve, they get a response at another region, the olfactory bulb, saying that there really is, they're able to get that information in. In that experiment, though, the animal's certainly not going to be making any sense of it about whether it's an odor or not. It's basically white noise. perhaps even more intriguing is Eric Holbrook did this. I think it was just five patients where they inserted some 3 electrodes right there at the olfactory epithelium, right adjacent to the cribriform plate, and were able to, in awake human patients, stimulate them, and at least some of those patients reported that they could perceive a smell. It's unclear, I would say, at this point whether that's true. They truly were getting a smell versus a tingling versus something else. These were all either normosmic or hyposmic individuals. No one was fully anosmic in these initial studies. But these are really proof of principle types of things. And one can imagine that going down the road, having basically a nasal prosthetic, a true electronic nose, might be an option for certain patients. This is probably the furthest away. Talking about the gene therapy, that's probably a 6 to 10 year road before you could get those into patients. The stem cell therapy, probably about the same. This might be a little bit longer, but it really depends on how the technology advances. So that's all I had prepared to say, but happy to take any other.
Great, thank you. I mean, we've got some very specific ones, Chris, sure. Super specific. But if you're going to sort of do your slight sort of crystal ball, at the moment, in terms of therapies which are available for people broadly with anosmia, hyposmia, et cetera, we're looking at sort of steroid treatments, et cetera. What's your sort of prediction for what will be the first new significant treatment that's going to come out of the work that you've been looking at?
My prediction is that gene therapy is going to be the first of those 3 categories to be able to get into the clinic. I think for a couple of reasons. One is that gene therapy is starting to come online for a variety of different conditions more readily. The more institutions are becoming comfortable with understanding how to safely deliver those genes and how to best do it. I think that's got the greatest promise. Now, of course, that's all. I'm not even touching on the possibility of having other drugs that might be identified, whether it's small molecules or biologics that could be useful as well. But of these types of more technology-driven therapies, I would say the gene therapy is probably most likely to come online first. But that's you never know what hurdles you're going to encounter.
Sure. Thank you. A question from Mike: is the part of the brain that processes smell trainable in the same way you have to train the brain to understand sounds when a cochlear implant's put in? I guess again we're very early.
We would expect so. Certainly, most odors that you encounter, you learn their meaning. You're not born knowing that a particular smell is pizza or coffee or anything else. You learn to associate a particular group of chemicals that elicit a particular pattern of nerve activity in your brain with an experience. The sight of the cup of coffee or the fact that you're walking by a coffee shop. So the fact that learning is constantly part of how we use our olfactory system, I would expect that the system is going to be well positioned to do that kind of learning in that kind of prosthetic training you would need.
Great, thank you. Chris had some really specific questions. 3 questions about the stem cell research. And I think that you and Chris are going to be the only ones who understand these. So
okay, I'll try to translate.
What is the lesion method in anosmic mice?
So in both of those cases that I showed you, both in the gene therapy story and the stem cell story, in both cases, those were basically knocking out, obliterating the ability of a single gene to function. So it's either deleting it completely or partially. In the case of the stem cell story from Brad Goldstein, what they did is something that's called an inducible one. So basically the gene's been manipulated in the genome of the mouse or in a line of mice, such that, but it's still intact until it is exposed to a particular chemical that turns on another gene that then creates an enzyme that can disrupt the gene that you're targeting. So it allows you to the mouse to grow up normally, and then you say, okay, today I'm going to destroy your sense of smell. And that's why that animal is, even though it's the same gene in the two studies, that animal is hyposmic versus the other one where the animal was born without the functional gene. And so it's got that more disruption. Well, it's not exactly born, but it's close to it. Yeah.
How comparable is it to human anosmia?
I mean, that's hard to say. We know that there are these single genes do cause anosmia in certain people, but it is in these cases they're both genetic changes, not something that you would acquire from a virus or head trauma or something like that. So the etiology, the cause is probably not similar to the majority of acquired anosmia, but but we do think there are probably some similar mechanisms in the basic ones.
And we've got quite a few questions come in, so maybe just a yes/no to this one from Chris. Do the implanted cells proliferate without exogenous growth factors?
I believe they do, yes. Although what I didn't show you is that one thing they could do that was really exciting is that they could put a few cells in a dish with various factors and turning on certain genes and create a bunch more that seem to be able to engraft, which would be the reason that would be really promising, is that you could have basically a pool of cells that you could use for multiple patients. If that
Fantastic. Works. Thank you. Avian says, how would you explain being fully anosmic but also having an acute sense of taste? Avian, I'm presuming, is taking on the tongue. I reason that the scents passed through my nose are still affecting or enriching my taste, but my brain doesn't know how to translate the aroma. What's your thought on that one? Sort of, I mean, there's always this sort of, I don't know if it's slightly like a pop science thing, whether, well, it's just saying that, how would you explain having a fully anosmic but also having an acute sense of taste?
Well, I mean, I guess that really depends on what you mean by taste. If you're using it the way we scientists would, where we're being very specific for sweet, salty, sour, bitter, umami, then as you described at the very beginning, those are two separate senses that only get put together in the brain to create flavor. So that would be if one is getting a more complex perception of food and drink that's beyond those 5 basic tastes and beyond the heat of chili peppers and the coolness of mint and that kind of stuff, that come from spices, that would suggest to me that there is some residual smell function, that it's severe hyposmia rather than complete anosmia. But that's
interesting. Thank you.
Possibility.
Coming through here, lots and lots here. Keith asking, has any work been done using biopsy in post-viral anosmia or hyposmia to compare the actual state of the epithelium and how it compares to normal?
There a limited amount, because as you can imagine that doing a biopsy, if it's not needed for, one wouldn't do a biopsy just for a scientific study or just for diagnosing post-viral anosmia. That would probably not pass ethical muster. So the only way to get them is if one is in there doing some other surgical manipulation or treatment. I know people are just starting to do this with COVID related, but most of that's been post-mortem that I know of. So.
Thank you. Smell training. You're still down as Wine O'Clock. I don't know what your real name is, but Wine O'Clock. Thank you for your question. Is trial training more appropriate as soon as the loss of smell occurs? I lost my sense of taste after a knock on the head 2 years ago. Is it still worth trying? So how does smell training as an approach, as a therapy, sort of sit in with the research you do in general?
I say the field varies in its confidence in smell training as a broad-based therapy. I think it's promising, especially in potentially in post-viral hyposmics, seems to have the best evidence supporting it. Although I think we're still at a point where we don't know to what extent it may be effective versus just spontaneous recovery. And I think the evidence, or what we know about duration, number of odors, all those types of things need to work. I'm real hesitant to provide any specific recommendation when it comes to smell training, because I think it's still too early days to really know that. That being said, it's something that should not have any negative side effects. So it's if someone wants to try it, there's no harm.
Good. Charlie, you put it very nicely in here, saying smell training never hurts to start and use as a mindfulness exercise, if nothing else. In that point, you sit and have a moment to. We also did a lot of work as well that. So we've got a couple of pieces of smell training for those who are interested in it. And one of the approaches is very much to work on the memories on the other side, and keeping those bring to light to help with the psychological impact. What else are there? Smell training on cures, help me, you may be right. I'm just seeing if there's anything else that I haven't picked up in here. I'm sorry if I'm doing la, just trying to get all the pieces. I think I've managed to pick up pretty well everything. If I have forgotten something or gone over it, please do let me know. Here we go. Yes, Avian. This with supporting cells. Avian, I'm not sure I understand what your question is. You said this with supporting cells, basal cells in the olfactory system. Is the principle the same for the smell?
So with the basal cells, if I think I understand it, if the basal cells contain different groups of cells and they're including the stem cells, and then cells that we call progenitor cells that have started on their way to their final identity. And so, but those initial stem cells give rise to the supporting cells, the sensory cells. We talked about the glandular cells that make the mucus, and other stem cells. They can regenerate themselves. So the stem cells have that potential to do all of those. As they mature and what we call differentiate, they become more focused on just one line or the other until they can only become neurons, so they can only become supporting cells. So I hope that answers the question.
Yeah, thank you. Question from Charlie, who is a congenital anosmic, saying, would the implant version be turn-offable? I'm terrified of being cured because I think it would be absolutely overwhelming. An adjustment period would be nice.
Yeah, those types of things are absolutely valid concerns that would have to be addressed. I mean, you certainly hear some stories now and again of someone who may have had a conductive issue with their sense of smell and not been able to smell for years, and then had some surgery, whether severe polyps or some other structural issue that's been resolved, and can smell again, and it being quite disorienting. And so, yes, having those types of things for people who are who use hearing aids, so we're not talking about cochlear implants, but just general hearing aids for amplification, there is a tuning process that everybody has to go through in order to optimize it for that individual. And most people who first get them find really disorienting and hard. You have to get through that initial stage. I would imagine that this is going to be a similar process.
Thank you. Rita, this is a question I had in mind as well. Thank you. What of this research is at the point of clinical trials or enrolling for clinical trials?
So at the moment, none.
Right.
I mean, I can tell you that each of them are in the discussions about how to get to clinical trials, but a lot of groundwork has to be laid before you can do the clinical trials, because we need to do that ethically and in a way that is as safe as we can make it for patients that are enrolling in that. Just like we've all seen with these vaccines over the last year, you would not want to participate in a trial if you knew that there hadn't been a lot of care taken by companies in creating safe things as safe as they could. So that's really where we are at the moment.
And I think that's a reminder as well that despite the fact we've only got 5 senses and that the rest of the senses have been so well researched, or at least the other 3, that this is still a new area of work coming through at this point to get up to those points. Keith asked a question back to mucus. I note that smell is restored temporarily after bike ride when my nose is streaming. Most of the time, a very dry nose and anosmia. Is the mucus doing this, or the whole epithelium being somehow stimulated?
That's interesting. I mean that's certainly and okay. Just the common disclaimer: I am not a physician, nor can I diagnose anybody over the edge. So not providing a diagnosis, but certainly when you have a restoration of smell or taste, for that matter, after a certain activity or certain condition or something, that may be a good clue about what is causing the underlying problem. And yes, one could imagine that if one possibility there is that there is an occlusion, there's a blockage of that because mucus being too thick or being prevented from flowing easily with polyps or something, where its odors are not able to get up to the olfactory epithelium. And then once you're in a situation where it can flow more freely, that clears things out and allows access again. That's one possibility that one might want to get investigated. But yeah, I mean, that to me, if you get function sometimes and don't get it others, and you have a rhyme or reason about when that happens, to me that's an important clue that you'd want to investigate with your physician.
Muted myself in the background. Let's just see. So I think we've asked this question lots of different ways about, what's the first treatment going to come, and are we going to, finger in the air? What's your time frame is our last sort of closing thought on this one, because of course everyone feels very kind of impatient to get involved. What's your sort of time frame, just to sort of set our expectations then?
Well, I think for the types of therapies we're talking about here, decade is probably a reasonable one because of all the regulatory steps one has to go through for getting these types of things. I must say though that, and I guess I mentioned it before, the prevalence of COVID-related smell loss is not only raised attention, which is important, but unfortunately it's also severely raised the numbers of people who are experiencing a smell or taste disorder. And all of those things are going to increase the pressure on funding agencies to devote resources to that. That doesn't only allow smell and taste scientists like myself to put more people on the projects and push things forward faster, but it also attracts people from other fields that might have really creative ideas to speed things along. Someone who's been working on gene therapy in the eye, for example, or working on stem cell treatments in skin, to turn their attention. And so it brings more people in to the field as well when there are resources that allow them to do that kind of research. So I am cautiously optimistic that we're about ready to see a real uptick in clinically focused smell and taste research.
Fantastic. I mean, once you get in those cross-fertilizations of ideas, then research can really take off. Well, thank you. We come right up to the end of time. Can I say on behalf of everybody on the call, please do pop in any message you have for Stephen. He'd love to read them as we're closing up. But thank you all so much for your time and your focus on this area. I think it really means a lot for people who, in many ways, you said about saying we can tell people you mentioned this earlier, we can tell people about the condition, but I think it's the experience of a lot of people with smell and taste loss or congenital loss that it's sometimes is hard to engage people in that. So that to know that you're there and your group of people there focusing so much on this area in an area that might not get a huge amount of funding or attention, and really to open up that space, I think it means a lot to know that you're there and you're doing that. So thank you in general for the work you do, and thank you for your time this evening. Lots of lovely messages coming in. Duncan, I think you wanted to say a few words as well.
Yes, thanks, Tom, and thank you very much, Steve. That was a brilliant. Thank you. And listen, I just wanted to say a huge thank you to you, Steve, on behalf of all of us at Fifth Sense for not just for this evening, but for all the work you've done on behalf of our community, the people we represent, and the support you've given to Fifth Sense over the years, which I really appreciate. And whilst COVID's been an incredibly difficult time for us all, it has prompted a lot of interest in smell and taste disorders, and it's going to be really interesting and exciting to see how we can work together to take particularly patient-focused research forwards. So here's to a bright future.
Well, thanks, Duncan. It's been a real privilege for me to work with Fifth Sense over the last few years, and I certainly hope that it's able to continue to grow. And again, another thing with we were talking before we got on, another silver lining from COVID, the fact that we are more attuned to doing these types of events where people, we got people from California to Australia, and it's at least somebody, I'm not doing the math here, but at least somebody is up in the middle of the night right now doing that. And that's great to be able to reach and provide information as people, and hopefully they can do the same to their networks.
Excellent. So thank you, Stephen. Thank you to everybody. Thanks for all of your questions and your thoughts you've brought through as well. Do keep an eye on your email. We'd love to know what you think about the session. We really want to know we're giving you what it is that you want. So keep an eye out. There's an evaluation email. It just takes a few moments to fill in, but it's hugely helpful to us. Keep an eye out for future events as well. Any ideas for future events you'd like to see us do, please do let us know. We are planning on continuing these. Lockdown restrictions lifted, be damned, we're going to carry on with our Zoom events, getting out, connecting everybody, because there's an immediacy to these sessions which I think we found incredibly valuable. So look forward to seeing you all again next time. If you're done, just click on leave session. Thank you all for being here.